Hey, it's Mr. Shrum. Welcome back to a biology walkthrough tutorial. And we're going to look at inheritance patterns and genetic research. We're going to use statistics and probability to calculate variation and distribution of expressed traits in a population. Does that sound difficult? Well, it's not that difficult. Let's get into it. By the end of the lesson, you'll understand everything to a T. But jumping into the warm-up, Gregor Mendel examined simple patterns of inheritance in his work with pea plants. Unlike pea plants, many human traits have complex patterns of inheritance. For example, some traits are controlled by several genes together. What is a trait that probably has a complex pattern of inheritance? Explain. So we have a sample answer. Human height probably has a complex pattern of inheritance because people in the same family vary in height. This difference in height suggests that multiple genes may control human height. And the next one, genetically modified organisms, GMOs, or organisms that have had their genetic code altered. Scientists use genetic modification to create crops that are resistant to diseases or certain temperatures. GMOs in our food supply have been a subject of great controversy. List 5 questions you have about GMO foods. Are genetically modified foods healthier than non-GMO foods? Does consuming GMO foods cause any potential harm? Can genetic modification allow crops to grow in areas where they normally wouldn't? What effects do GMOs have on native organisms? Do genetically modified crops have any positive or negative effects on the environment? All very good questions. Let's get into it. Genetic linkage. All right, so you're one in a million. Have you ever heard someone say that? Actually, from a purely genetic standpoint, every human is one in trillions. And to understand this fact, we need to understand some of the key relationships and combinations found in our genetic code. Genetic recombination and crossing over. Have you ever wondered why a close relative looks so different from you? Even though you both come from the same family. Individuals may look similar, but no two people are exactly the same. That's because our DNA genetic code undergoes many permutations and combinations to yield different characteristics that make us unique. The parental DNA follows a set of mechanisms to produce new DNA that is passed to the child. This mechanism is known as genetic recombination, and that occurs in different ways, one of which is the crossing over of chromosomes during meiosis. Let's see how the genetic code undergoes such combinations. So here we have a graphic of meiosis, prophase, metaphase, anaphase, telophase, and cytokinesis. Chromosomes are packaged structures of DNA that contain genes. Genes are present at specific places called loci. And just to check my pronunciation. Always double check. Loci, my bad. Loci on the chromosome. During meiosis, segments of the chromosome containing these loci cross their place with the same loci of another chromosome. The genes present on these loci are exchanged, resulting in a chromosome that has different genetic makeup than the original one. This process is called crossing over. The two chromosomes with the changed genetic content are called recombinant chromosomes. A chromosome crosses over only with its homologous counterpart. Homologous chromosomes contain the same genes in the same loci, but they have slightly different DNA sequences. The breakpoints, or places where the exchange occurs, can be anywhere on the chromosome. The process of crossing over increases genetic variation in species because different combinations can be made. Genetic recombination. Genetic recombination produces a number of new genetic recombinations by independent assortment and the crossing over of chromosomes. Genes usually undergo independent assortment during meiosis. Independent assortment of alleles means that alleles distribute themselves randomly during gamete formation. That is the selection of an allele for a particular gene by a gamete is independent of the allele for any other gene. Independent assortment for a species can be calculated using the formula 2 to the n power. And that n equals the number of chromosome pairs in that particular species. Gregor Mendel, the father of modern genetics, used pea plants to study genetic traits. There are 7 chromosome pairs in a pea plant. The formula 2 to the n power reveals the possible genetic combinations that a single gamete of pea plant can produce. 2 to the 7th power equals 128 combinations. We can then find the possible genetic combinations of a zygote formed from 2 parent plants. You have 128 in one and 128 combinations of the second plant. Multiply that together, you get 16,384 combinations. Humans have even more pairs of chromosomes. We have 23 pairs. The independent assortment will be about 2 to the 23 power times itself. 2 to the 23 times 2 to the 23rd power, which is around 70 trillion. These figures calculate only the independent assortment possibilities, not possibilities from genetic recombination. When you also consider the different methods for recombination in a species, the genetic possibilities are nearly endless. And the number of chromosomes in the cell of an organism is called its karyotype, the human karyotype. So, to check your understanding, fruit flies have 8 pairs of chromosomes. Calculate the genetic possibilities from independent assortment of a zygote made from 2 fruit fly parents. Show your work and the formula you used. So we have 2 to the n power, right? Here we're told it's 8. N is 8. 2 times 2 to the 8th power. Oops, sorry. 2 to the 8th power. And if you plug that into the calculator, you get 256. That is the independent assortment combinations. And then you multiply 256 times 256 because it takes 2 fruit fly parents. So 256 is just for one parent. This is for 2. And you get 65,536 combinations. Linked genes. Chromosomes contain genes. They are located at different positions on the chromosomes. Some of these genes may lie close to each other, while others may be far away from each other. Certain genes are located on different chromosomes. The genes that lie closer are called linked genes, and the ones on different chromosomes are called, well, unlinked genes. We saw during crossing over, that genes are exchanged. Linked genes have a higher probability of traveling together than unlinked genes. Genes and genes located farther apart on the same chromosome. So if they're linked and close together, they're more likely to travel with each other. However, a linked gene, linked genes do not always travel together. They can be separated during crossing over. So identify how strongly linked gene 1 and gene 2 are on each of the chromosome models. So, okay, here. Check it out. We have gene 1 and gene 2 on the same chromosome and very close together, strongly linked. Here we have gene 1 on one chromosome, gene 2 on another. That's referred to as unlinked. Then we have gene 1 and gene 2 on the same chromosome, but farther apart than this one. So it's linked, but not very strongly. All right, I think that's a good stopping point. And next time we're going to look at patterns of inheritance. Until next time, have a good one, and I will see you all later. Bye-bye.